and go with glucose to pyruvate. We broke a bond here. What happens when you break a bond?
Energy.
Energy gets released. Right? So we're going to absorb that energy and use it to make ATP. We're going to make actually 4 of them. Right? So we get enough energy from breaking that bond to take 4 ADPs and turn them back into 4 ATPs. Okay? But there's a catch. Remember, every reaction has activation energy. You got to put energy in to make this whole thing start. So you actually have to burn up 2 ATPs to get glycolysis going. So you start in the hole. You're in -2. Then you make 4 more using the energy from breaking that bond. So your actual net gain plus 2. So that's the number I want you to remember, a net of 2 ATP for glycolysis. And that means we netted 2 ADPs, 2 ATPs. And the energy to make that happen is substrate level phosphorylation because we use the energy from breaking that bond.
So it's 2?
It's 2 ATP, substrate level. We're also, at the same time, going to make 2 NADHs. So that, again, means 2 NAD pluses, picked up 2 hydrogens from this glucose, and became NADHs. So there's the picture, showing you pyruvates being separated, reminding you that substrate level phosphorylation was used to make the ATP.
Wait, the electron carriers, are those the NAD?
NAD pluses, 2 of them become NADHs. And all that's exactly the same in bacteria as it is in people. Other questions on glycolysis? Okay. I'm glad we had time to get to this one. Transition step, if you have had other biology classes where you've learned this before, you've never seen the words transition step before. Okay, your microbiology book is the only one I've seen that uses this term. It's not wrong, but they're just being a little bit more precise. Okay? Most books call this stuff pre-Krebs cycle, the things you have to do before the Krebs cycle can happen. Okay, your book calls it transition step, and I kind of like it, giving it its own name. So we're going to go with what your book says is the transition step. Just again, a note for the online class that you're not going to find transition step in your anatomy book or in your general biology book.
And this is the one that's also called citric acid cycle, right?
This is not the citric acid cycle.
So in this case, it will actually be Krebs. I mean, after we get
Krebs
past
has
this,
3 names.
right?
So we'll wait till we get to Krebs
Okay,
to ask that question. Okay, so this is before any of that happens.
Good.
Okay. All right, so we're still in the cytoplasm for bacteria and for humans. We're going to take those 2 pyruvates that we just made, and there's a picture of this on your next slide, and we're going to cut 1 carbon off of each one. Right, we're going to break 1 carbon off of each. So now we're left with a chain of 2 carbons, and that molecule was our molecule out, acetyl-CoA. So that's our molecule out. Even though we break bonds here, energy gets released, we're not 100% efficient, we don't absorb that energy to make ATP out of it. That energy is lost as heat. Okay, so no ATP from this step, therefore no phosphorylation happens. But we do make 2 NADHs.
Right.
Two more NAD pluses pick up those electrons or protons and become NADHs. Okay, so again, if you go back to your A&P or your general biology book, this is going to be called pre-Krebs cycle or pre-citric acid cycle.
Okay.
But your book calls it transition step. And there's the picture I promised you, showing you the carbons being cut off. All right, so what happens to those carbons that we cut off with pyruvate?
They release energy?
Well, energy is released, but we don't get to absorb it, right? That gets lost as heat. Yeah. Those carbons are going to be used to make carbon dioxide using some oxygen from the glucose. Remember, we were ignoring the oxygens, the C6H12O6. Some of those O's are used to make carbon dioxide here. This is a waste product. for us as humans and for bacteria.
A molecule out is also for glycolysis, two
Well,
carbon
this is-
dioxide molecules.
No, this is transition step, not
Right.
glycolysis. I wouldn't call them a molecule out because they're waste. This acetyl-CoA is a molecule out because they're gonna use it for something else in the next step. Yeah, CO2 is a waste. For bacteria, it just moves out by diffusion. It's gonna diffuse right out of the cell and go away. For us, we collect it in our bloodstream, carry it to our lungs, and exhale it. And so waste product for everybody there.
CO2 is waste?
CO2 is waste. Yeah, we don't do anything with it, but breathe it out. And again, bacteria leave it by diffusion. Well, anytime. So now we get to Jamel's question about what we call the next step. Okay, I learned it as Krebs cycle, so that's how I'm gonna say it. That's also what the general biology book will call it, the Krebs cycle. So if you've had Bio 101, that's how you learned it. If you've had A&P, that book calls it the citric acid cycle. And your book calls it the TCA cycle, tricarboxylic acid cycle.
I'm not gonna work.
Has three names for the same thing, Krebs, citric acid cycle, and TCA cycle.
But you're using,
For the last one, was there phosphorylation?
She's not okay.
No, there was no phosphorylation in the last one. It's gonna come out of my mouth, the Krebs cycle. I try really hard on tests to call it the TCA cycle because I know that's what your book calls it. But you should know all three names because depending on the class you're in and who's teaching you,
On the tenth, you're gonna call it a try.
I'm gonna try really hard to
No.
call it the TCA cycle.
so TCA,
I thought it should just be
crab,
Krebs.
and citric acid.
I mean, that's really the name you taught me first, but then I like the last name.
Depends on the book you read, three names for the same process. Krebs, just to give you some idea of where this came from, Krebs is the last name of the guy who figured out how this works. So some books give him the credit for it. Citric acid and tricarboxylic acid are two of the intermediates involved in the step. All right, so no matter what you call it, okay, what happens with those two acetyl-CoAs that we just made in the transition step? Any idea what we're gonna do to those carbons? Yeah, what we've been doing all the way through here. Breaking them, right? We're gonna break the bonds, separating them from each other. So we're gonna have individual carbons, which are gonna join with oxygens to make carbon dioxide, which is still waste, right? We don't do anything with it, bacteria don't do anything with it. It's gonna leave bacteria by diffusion, it's gonna leave us when we exhale. So that's not our molecule out, okay, that's a waste product.
Right.
And to me, really, that's the coolest part of metabolism, because this morning I had solid food for breakfast, right? I had oatmeal, I had grapes, I had to chew, right? Solid stuff, and watch. There it goes. Right? All of the carbon from the sugars in my grapes and in my oatmeal, because I definitely can't eat oatmeal without sugar. Right? All of the carbons from that sugar get converted into a gas, which we exhale. The calories, of course, stay with me, but the carbon goes away. I just think that's kind of neat. Solid food goes to gas. So since that's not the molecule out, that's waste products being gotten rid of, the molecules out are actually the electron carriers, because they're gonna be what goes on to the last step. They're what moves on from this. So, you just draw yourself a little arrow, the electron carriers, there's a lot of them. 6 NADHs and 2 FADHs are produced in this step. And again, remember, that means 6 NAD pluses picked up hydrogens to become 6 NADHs. 2 FAD pluses picked up hydrogens from these acetyl-CoAs to become FADHs.
So we have 2 NADH,
6 NADH.
I mean 6 NADH and
And
2
2 FADH. Mhm. We do make a little ATP at this step. We broke bonds. Break bonds, energy gets released. We are able to absorb it at this step and use it to make 2 ATP. Again, substrate level phosphorylation because we're using the energy from breaking bonds, exergonic reactions. And picture that on the next slide, like the carbons breaking apart. Questions?
Is ATP used to,
Not for any of the other steps. Glycolysis is the only one where we go into the hole. Good question.
That's the first one.
So at this this point, the glucose that we started with, we have chopped up every single carbon off of that glucose. Has been reduced to carbon dioxide, right? We add them all up, all the CO2s on the board. We made 6 CO2s from the 6 carbons in glucose. We have completely broken down the sugar molecule to the central metabolic pathways. So the only things we have left to go on to the last step are all of those electron carriers that we've been generating. So, that last step, your book calls respiration. I like it, let's go with respiration. I put the other term up here because some of the general biology books call it electron transfer phosphorylation. Respiration's so much easier, let's just call it respiration. Okay? That'll be the term that we'll use. Location here is where we get a difference. We are in the mitochondria for eukaryotic cells. Okay, so if you've had general biology, you know what that is. If you haven't, I'll draw it for you. Bacteria don't have any internal compartments, right? No nucleus, nothing with a membrane around it. Okay, so bacteria, blank slate. Eukaryotic cells, you know, have the nucleus in the middle, membrane-bound compartment to hold the DNA. They've got lots of other compartments inside of them. This one is the mitochondria. It's got 2 membranes. Okay, so if I draw it bigger, just so you can see, the outer membrane is nice and smooth. The inner membrane is folded up, kind of like pleats on a skirt, right? When you fold stuff up like my sleeves, there's more fabric there, right? If I pulled all of those little pleats out, the sleeves would be like this long, right? So we can fit more inner membrane in by folding it up than if we had had, 2 smooth membranes. The important part for this is that there's a space between the inner and outer membrane. So that's where we are inside of a eukaryotic cell. Bacteria don't have mitochondria, so the membrane that we're going to use here is the cell membrane. We're doing this right on the edge. We're not doing the cell membrane of eukaryotes. We're going to use the membrane inside of the mitochondria. So big location difference here. Molecule in, all of those electron carriers. So if you go back and count it up, we made 2 NADHs in glycolysis, 2 in the transition step. We just made 6 from the TCA/Krebs cycle, and 2 FADHs from the TCA/Krebs cycle. So all of those are coming over to this step called respiration. The molecule out is ATP. We get most of our ATP from this step. Because if you go back to everything on that side of the board, we made 2 little ATP right here in glycolysis, and we make 2 ATP in the TCA cycle, and that's all we've made so far. From 1 sugar is 4 little measly ATP. So we're going to get massive amounts, 32 to 34, out of this step as our molecule out. And they're going to be made by oxidative phosphorylation this time. Not by breaking bonds like we've been doing here, but by using the energy from proton mode force, having negative on one side, positive on the other. So everybody's copying, have time? Yes, sir.
Never mind.
Okay. So, the electron transport chain. Here's where we get to talk about how the one side of the membrane became negative and the other side became positive. Let me get rid of glycolysis and transition and Krebs here. All right. So, here's our membrane. We're talking about a bacteria. This is the cell membrane, right? We're talking about a eukaryotic cell. It's the inner membrane of the mitochondria, the one on the inside, that ruffly one. The electron transport chain is a series of proteins embedded in that membrane. Electron transport chain, series of proteins embedded in the membrane. And what it's going to do, we use it to generate that proton motive force. So all of the NADHs and all of the FADHs that we've collected in the previous steps are bringing their electrons over to this membrane.
This is the
Integral?
mitochondria.
For eukaryotes, it's the inner membrane of mitochondria. For bacteria, it's the cell membrane.
Okay, that's only for bacteria, integral, okay.
Yep. Cell membrane, bacteria only.
Prokaryotic.
Eukaryotes don't do this on their cell membrane.